Good afternoon, everybody. Thank you for joining us today in this webinar. I'm Sangya Pundir. I am the User Experience Manager for UniProt, and I'll be taking you through a few useful tips and hints and a case study for looking at disease models. So, looking at exploring models for human disease using UniProt today, what we'll do is go through a few general areas of the website that are useful if you're working with disease information, and then looking at this specific use case as well. So for those of you who might be new to UniProt, I'll start with a short bit of introduction. UniProt is a universal protein resource, and we aim to provide the highest quality, most comprehensive, and thoroughly annotated protein resource freely to the scientific community, that is yourselves, to use. We have a sort of three-pronged approach to fulfilling our aim. The first one is to provide detailed information on biological aspects of as many proteins as we can, such as function, interactions, pathways, subcellular location, disease involvement, and so on. We have an element of expert curation that goes into the UniProt knowledge base, which helps us provide all of this information in a detailed manner. The second aim that you can see on the screen is to do with creating and maintaining an archive of sequences for proteins, including different forms of these sequences, such as isoforms, sequences with variants, with post-translational modifications, and so on. And we also aim to maintain stable identifiers for proteins to help you come back to your proteins of interest and find them easily over time, and these are known as protein accessions, and they're found in every UniProt entry, and they stay the same from release to release. So that's just a very brief introduction to what UniProt is and what we do. Looking at the resources we provide, one of the main activities that our users perform on UniProt is searching for proteins and protein-related data. On the screen, you can see a number of boxes with different data sets written in them. These are the data sets that you can search in UniProt. The one that's searched most commonly that you're most likely to look for is UniProt Knowledge Base, or UniProt KB, which is the blue box on the left of the screen now. The UniProt knowledge base contains all of the protein records that have the information about function, sequences, interactions, and so on compiled within them. There are two types of proteins in UniProt knowledge base, two sections of the knowledge base. The first one, as you can see there, is SwissProt, which contains manually annotated and reviewed proteins. These have undergone expert curation with our curators. And they've added in all of the information that they compile from literature analysis, their own analysis tools, including sequence analysis, and so on. The second section in the knowledge base is Tremble, and this section is automatically annotated based on rules and machine learning approaches that learn from the information in the SwissProt section, but is not reviewed by the UniProt curators. There's a number of other data sets that you can see on the screen, which can also be searched in UniProt. The one that I'll just draw your attention to briefly now is the diseases data set under supporting data, which is the purple box, as you can see. We will look at this in a bit more detail in the following slides. We also have 4 tools as part of UniProt, and these are noted at the bottom of the screen. So we have a BLAST sequence similarity search tool. We provide sequence alignment tool, ID mapping, and a peptide search tool. So if you're working on disease-related research or looking at variant information at the protein or gene level, when would you come to UniProt? What data can you come to us with? So you can look up variants in UniProt and learn about their relationship with a protein's function and their role in linking that protein to diseases. To do this, you can come to UniProt and start with a disease that you're interested in, or you can start with a gene or a protein that you already know you're interested in, which might be related to diseases. Or if you have a variant identifier, such as a DB-SNP identifier, and you're interested in a particular variant, you can search with this in UniProt as well. So there's these 3 input points coming into UniProt that might be useful for you. We'll very briefly look at each one now. So let's say you're interested in exploring a particular disease in UniProt, since we're talking about disease models today. The best one of the ways that you could start is by searching for the disease itself rather than searching with proteins or gene names. To do this, if you look at the UniProt search bar, there's a drop down to the left of it that I've highlighted in red on your screens now. This drop-down shows you the data set that you're currently searching, and it's on UniProt knowledge base by default. But if you click on it, you can see that there is an option inside under supporting data to change your data set to human diseases. So if you had a disease in mind that you're interested in, this is one way you could start. You could select human diseases and type in your disease name, as I have done, and then hitting the search button will bring you to a results page like this. In this example, I've just entered in Alzheimer's. That's the disease I'm interested in. And as you can see, the results bring me descriptions of diseases that are cataloged in UniProt. So I can start by selecting the right disease. In my list now, let's say, for example, that I was interested in Alzheimer's disease, which is the second hit in this list. I can see a description of the disease, so I can decide that it is the right one for me, the one that I'm interested in researching. And underneath that, I can see a link to one UniProt knowledge base record. This is one record in my example at the moment, but it could be more. If you search for different types of cancers, this link here would bring you to all of the UniProt knowledge base proteins that are involved in that disease. So this is an easy way of finding the exact disease you're after and pulling out all of the proteins that are involved in that disease and starting your research that way. Taking a different approach, maybe you're researching diseases, but you already know the protein or proteins that you're interested in, and those are the ones you want to focus on. In this case, you will want to keep your search focus on UniProt knowledge base, which is the default in the search bar, and just enter in the names of the protein or the name of the gene that you're interested in. So let's say, for example, I've searched here for insulin, and you can see that I've got a results page here with various columns of information and filters on the left-hand side. So a useful thing to do here, if you know already that you're interested in disease relationships for the proteins you're looking at, is to customize the data that you're looking at in your table. You can actually change the columns that you're seeing here to see the information that you are more interested in straight away without having to go into every individual record. To do this, click on the columns button, and it will bring you to this page where you can customize the columns that you see in your results table. As you can see, there are many columns you can add to your table in UniProt. There's essentially a column for every type of information that is present for a protein in UniProt. So the easy way to do this is to use this search bar to type in a word or a phrase that you're interested in. So I've just started with disease here, and it brings up the suggestion of a column called involvement in disease, which sounds just right for what I'm looking for. So if you wanted to save this, you can just click on this. It'll be added to your column display, and then just click on the save button, which will bring you back to your results table, which is now customized for you, and in my case looks a bit like what you're seeing on your screens now. So as you can see, along with every entry that's come back in my results, so the first one being insulin receptor, there's now an involvement in disease column, which shows me straight away all the diseases that each protein is involved in. So I could now decide to look into a protein in more detail, or perhaps from here go into a disease I'm interested in and carry on that way. So these are sort of two approaches of starting in UniProt to look for the disease you're interested in. Let's now look at what you would explore on a single protein page if you were interested in disease relationships and variant data. So if you went to a UniProt knowledge base entry, if you clicked on a result, you would come to an entry page that looks a bit like this. I'm using a breast cancer related protein in this example, but essentially we follow the same template on all our entry pages, and you will find right on top of the page the protein accession, the stable accession that I spoke of earlier, the entry name, which is just a quick handy way of identifying your entry, a number of tools that you can use or formats that you can look at this entry in or download it in. And then you've got a little bit of an introduction about the protein, including protein name, the gene name, the organism that it comes from, the status of this entry, whether it's reviewed or not, and so on. So the information in the entry is in a document form. You can scroll as you read it, and it's divided in various sections, the first of which is the protein function, as you can see on the screen now. But as you might notice on the left-hand side, there is a navigation bar which shows you all of the sections, and one of them is pathology and biotech. This is highlighted in red on my screen now, and this is the one that will tell you about the disease involvement of this protein and any related natural variation that we know about. So let's look at the pathology and biotech section in a bit more detail now. It looks a bit like what you've got on your screens now. So we start with the involvement in disease comment, as is known in UniProt. And in this case, it's a breast cancer protein that I'm looking at. So you see a description of the disease that is involved in right there. And under that, you see a table of natural variants, which is mutations that link this protein to this disease. So all of these variants are related to this disease. If this protein was involved in multiple diseases, you would see the same pattern listing each individual disease one under the other with its related natural variants. In the natural variant table, you've got the position of the variant, what it changes the amino acid to, and any publications attached to it. So you might notice that there's publication tags, these yellow tags for each of the natural variants, which brings me to an important point to note, which is that in this table you see variants that have been expertly curated into UniProt by one of our curators, variants that have experimental evidence for being associated to disease or not. But there are more variants than this in UniProt. We import large-scale variants that are not experimentally proven, but might be useful nonetheless. To see those, you need to take a slightly different approach. So if we go back to looking at what the entry looks like, it looks like this now. On the left-hand side, there is a link under the display section which says Feature Viewer. This link will take you to a graphical view of all sequence features such as domains, post-translational modifications, and variants. And this graphical view will provide the data that is manually created, that's based on experiments, such as from the table I just showed you from the pathology section. But it will also show data imported from large-scale studies, which is not curated from experimental evidence. So let's look at the feature viewer. It looks a bit like this. This is an example of a feature viewer for a protein entry. And you can see there's a ruler on top. It shows you the full length of the sequence. This is 770 amino acids long. You can use this ruler to zoom in or drag your area of focus around. And then the actual sequence features are annotated using different shapes and colors, and there's a key on the left that shows you what type of features is in a track that you're looking at. This is a track-based approach, like many genome browsers you've probably looked at before as well. And right at the bottom, there's variants. And this track shows a graph of the number of variants at each position. So you can quickly see areas of high variation and areas of low variation in the sequence. This is an overview of variants for this sequence. If you clicked on this section, if you clicked on any of these sections, you can expand them to see more detail. And I'm going to show you now what the variant section looks like here. So as you can see now, we see an expanded view of the actual variants annotated on this chart. And what we're doing essentially is for every position, every variant that we know about is represented by a dot that corresponds to the amino acid that this position is changing to. So for example, if I click on the red one here, it brings up a pop-up which tells me that this is position number 693. This is a variant that changes the amino acid E to a G, which is why it's in the line corresponding to G. And there's a cross-reference and disease information related to this variant. You also, you will also note that there are different colors of these variants, and the key to these colors is on the left-hand side on your screen now. So the variants that have been reviewed and are known to be involved with diseases are the ones that are red. You can filter to see just these by just clicking on the red box here. All of the blue variants come from predictions, so they are imported from large-scale studies such as the 1000 Genomes Project and so on. And then variants that have been reviewed and are known to not be involved in diseases are colored green. The red and the green ones are those that are visible in the table that I showed you in the pathology section directly on the entry, and the imported ones that are not experimentally verified yet are not shown in that table. But here in this view, you can see them all together. There's also some yellow dots, as you might see, and these are these are the a different type of variant. All of the other ones are missense variants essentially, but the yellow ones are the stop loss, stop gain, or initiator codon related variants, and they're on their own line right at the bottom. So this is the variant view, and you can quickly see, just looking at the red and the green ones, the expertly curated variants from the view you saw earlier, as well as the additional ones along with them. So having looked at generally how to explore disease and variant related data in UniProt, we now come to the question in particular that we want to explore in today's webinar, and this is finding disease models for proteins in UniProt. And we'll follow a case study along with me now. So I want to take an example of a paper. There's a screenshot of just the title of the paper on your screens now. This is a yeast-based assay identifies drugs active against human mitochondrial disorders. So you're looking at a paper about the disease NARP. You don't have to read the paper yet, so I'll give you a quick introduction. So this is about the disease NARP, N-A-R-P, that's the acronym, and it mentions an assay they developed based in yeast. So they used a yeast disease model for the human protein that is involved in this disease. And let's say that you wanted to find a model for this disease in C. elegans instead of yeast, so that you could also do an experiment similar to the one that you're reading about in this paper now. So this brings us to our question of finding a disease model for a protein involved in the NARP disease, but in C. elegans instead of human. And how would you do this? How would you find and identify the right protein to carry out your experiment with? So we start by looking at the protein, the human protein that we are looking for a model for. So the paper lists some common mutations in the NARP patients in a gene encoding for a protein called ATP synthase subunit A. So this is a protein they've identified as being involved in the disease NARP that we're looking at, NARP. So we start first by looking for the human protein that is mentioned here, and I do that by just searching for ATP synthase subunit A in the UniProt knowledge base search bar right there, as you can see in the screenshot. So it brings me to a search results page that looks like this. You might notice that I already have the involvement in disease column in this view, as I customized my results page earlier. So looking at these results now, I was looking at ATP synthase subunit A, and I know I'm looking for proteins involved in the disease NARP. So the second result looks immediately interesting, as I can see that its name matches the one in the paper, ATP synthase subunit A, and it's involved in the neuropathy disease NARP that I'm looking at as well. So I zoom in on this and I click on the entry link to go look at this protein in more detail. So looking at the protein, the question that I'm looking to answer now is, can I find a variant on this protein's record that shows that there is a mutation that's linking this protein to the disease that I'm looking at? So I scroll down to the pathology and biotech section. I've taken a screenshot of that section directly. If you're starting from the top of the entry, you can click on the navigation bar on the left to jump to this section straight away. And I see that it's involved in the disease NARP, as I saw on the results page already. And there is one variant that is linked to this disease for this protein. So I see the natural variant here, and I see that there are 2 publications related to it, and that it corresponds to a DB SNP ID. You can go look at it in these other databases if you like. The links are to DB SNP, Ensemble, and ClinVar. But the thing that's interesting to me is the position. That's the bit that I want to now remember when I proceed with trying to find a model for this protein related to the disease. So this variant is at position 156, and it's changing the amino acid L to R, the leucine to arginine, at position 156. So this is information that I now keep in my mind as I proceed with my investigation. So it looks like I found the right human protein. It is involved in the right disease, and it has a natural variant as the paper describes. which links the protein to the disease. So I have found the right protein in the human organism, and now I'm looking to find a model for this. So the paper uses a yeast model. I think so. I'll just correct myself. The slide says I'm looking for a corresponding gene in yeast, but the paper has already tried the experiment in yeast. So we want to find a corresponding model in C. elegans. That's the organism that, let's say, you have access to in your lab. That's the one you want to do your experiment in. So I'm just correcting myself on the slide here. The paper uses yeast, but we want to see if we can find something in a worm. So I go back to the top of the entry here now, my human protein entry that I've identified, and I click on the blast button, change my focus to advanced blast because I want to play around with the options, and click on the go button. And that brings me to a page that looks like this. So seeing as I already know the type of organism I want my model to be from, and in my case I've chosen C. elegans, and the paper had chosen yeast. Seeing as I already know this, I click on the target database drop down to change my selection from UniProt knowledge base, which is the default, to nematoda in my case. And of course, if you were doing what the paper did and just reproducing that, you would choose fungi, for example. So you can narrow down your blast search set in this way. So I choose nematoda, run my blast, and I come to the results, which look like this. So looking at my results, I can see I've got results from different organisms, and on the left there is a filter called popular organisms. I see C. elegans is in there, and I click on that to just narrow down my search results to the organism I'm interested in. And I repeat again, you can ignore the yeast in the title that links to the paper, but I'm looking for a C. elegans model now. So that quickly narrows down my results to just the organism I'm interested in, and I can see from the identity scores that the top few results look to have the same identity score, 31.2%, but one of them is a SwissProt entry, as I can see for this golden icon, and that stands out for me because I know that a SwissProt entry is expertly curated and is likely to have more annotation in it. So I click on the SwissProt entry to go and investigate it further. So when I do that, what I'm looking for is to see if the variant that links the human protein to the disease that I'm interested in now is also present in the model that I'm now investigating, in my case for C. elegans, or in the paper's case they must have done it for yeast. But if the amino acid that mutates to link the protein to the disease is conserved in my organism that I'm now investigating, then that is potentially a model for this disease. So I look at the alignment here, and I select on the left hand side. You can see there's a number of options for highlighting certain things directly on the alignment to help you look for certain conserved annotations, and I select natural variant here to help me narrow down to what I'm after. So if you remember from a few slides ago, when we looked at the human protein that was linked to the disease, it had a variant at position 156, and it was changing the amino acid L to R. So I kind of look around that region. This is the human sequence here, and I can see that there is a natural variant highlighted on an L, which is around 156, which is 156. And I can see that it's conserved in the sequence that it was aligned with, which is from my C. elegans protein. And not only that, there is a variant annotated on the amino acid before L, which is an A, and that is also conserved. So both of these amino acids that are interesting to me are conserved in the C. elegans protein as well. And that says to me that this is a potential model for this protein, and has answered my question of whether I found the right model for what I was looking at. So if I just summarize the results, we started with the human ATP synthase subunit A protein, and we found a potential C. elegans protein that had exactly the same amino acids conserved, which lead to the disease in the human protein, which means that they are a potential model for this disease. The original paper used a yeast model. So what I can do now is try and compare all three proteins. So I take the ATP6 human, which is the one I found in my initial search. I take the ATP6 yeast, which you can find following same steps. You can do a blast against yeast. And then I take the ATP6 in C. elegans that I found using my investigation now that I've walked you through. And I take these three and align them to see what similarities I can find in them. And this is the results of the alignment here now. I highlight the natural variant annotation again, so I can easily compare the particular variant that I was after, which was, if you remember, in the human protein at position 156. I'm just going to highlight it now, counting back from 149. That's where it falls. I've highlighted it in red. And as you can see, in the human variant, there's an annotation for natural variants at the position for A and L here, and we know that it's the one for L that links this protein to the disease, and the L is conserved right throughout all three sequences. You can find it in the C. elegans one and also in the yeast one from the original paper. And then it may or may not prove to be relevant, but it's interesting also that the amino acid just before L also has a variant annotated on it. This is the A, and that's also conserved in the C. elegans protein. But not in the yeast, but L is the critical one, and that's conserved in all three. So this analysis tells me that I could potentially use this C. elegans protein in a similar way that the original paper used the yeast protein as a model for this human disease involved, this human protein involved in this disease related to neuropathy. Another interesting point here is that you might notice that the kind of short form entry names for these three proteins are very similar. There's ATP6_human, ATP6_ C. elegans, ATP6_ yeast. And that's by design. So in UniProt, in the UniProt knowledge base, whenever a protein is curated, if it seems to be similar enough to a protein in another organism through blast analysis or other such methods, we try to give it the same entry name, the same protein name to make it easier for you to analyze. But if you follow these steps, you can identify proteins even from the Tremble unreviewed section that may or may not have the same name, but as long as you've checked the sequence similarity yourself and checked for the key positions that need to be conserved for your investigation, then you can be sure that you found an interesting protein that you can use as a model for a disease. So that was just a quick walkthrough of how you can start with a human disease-related protein and find a model for it from a different organism. And we did this by using various UniProt entry related tools and so on. So we used the UniProt knowledge base search. We customized our results to always see the disease section to make our analysis quicker. We looked at the entry view, and we also looked at the feature viewer. We ran a blast sequence similarity search, and we used the alignment functionality to help us come to our conclusions. That's what I wanted to show you today, an example of how you can do this with UniProt, and also an introduction of different ways of accessing disease-related data in UniProt. But if you try some of this on your own, and if you have questions about UniProt data or functionality later, you can always write to us at help@uniprot.org as well. Just quickly before I end this webinar, I wanted to acknowledge the UniProt Consortium. There's a big team that all work very hard for making UniProt possible. There's people across 3 sites. We have the EMBL-EBI in Kingston, UK. We have the PIR in Washington, DC, and Delaware in the US, and we have SIB in Geneva in Switzerland, and there's various people involved from all 3 sites. That's it for today. Thank you for listening. If you do have any more questions, feel free to write to us at help@uniprot.org at any time. We're always there to try and answer your questions and help out in any way we can. We also have a Twitter account. If you want to follow us there, we're just at UniProt. We're also on Facebook. So you have various ways of getting in touch with us. If you'd like to send us your suggestions for webinars and your feedback or any other type of question, you're most welcome. Thank you very much for listening. We'll be closing this webinar for today. We really enjoyed sharing this information with you. We hope it was useful for you as well. And please feel free to connect with us on our other channels. Thank you very much and have a nice day.